Tonight we're going to be looking at evolution from an engineer's point of view. I have a multi-decade career in engineering, but in the last 10 years I've been relearning biology. It's been helped by my wife, who is a biostatistician, and she is making sure that what I'm saying and putting together is technically correct. What I've found is there's about 2 million active biologists right now collecting data. That means the amount of publications in biology is intensive. It's an incredible amount. It's really hard to get a picture of all of it. You need to specialize. And I think most people in biology are specialized in a small area. They're digging deeper and deeper. Also, I would say most of these are not staunch evolutionists. What they're developing fits in the evolutionary framework and they just go along with it. But I don't think they are really promoting a evolution point of view, a worldview. But there's some of them. As you look into what's being developed now, you'll find there's areas of complexity, details that just don't fit the theory of evolution, which is a very high level, broad approach to that. My wife had developed a calculation, a model of that, and we're going to include that because it helps to look at some of the things that normal evolution, survival of the fittest, can't explain. There's just too much complexity in there. If you've ever looked at flat fractal geometry, you realize that as you look finer and finer, there's still more and more details. You're just going down deeper and deeper. So I think there's some serious problems in evolutionary theory, and some other people do as well. Yet there are people who strongly support evolution as the only solution. And in fact, some of them are very adamant. Several years ago, I came across this site called Rational Wiki, and I'm going to show you that site in a quote. At the bottom, you see the reference there to Rational Wiki. They have something called the Salem hypothesis at that link. And it is the observation of the apparent correlation between the engineering trade and creationist beliefs, possibly due to crank magnetism or some other crackpot idea. It holds that people who claim science expertise whilst advocating creationism tend to be formally trained as engineers. In other words, they're saying those who don't believe in Darwinian common descent are crackpots. And I would argue they are really missing the point. They have really not really looked at the details out there. They do not understand that the people who look at an engineering view of biology are seeing things that they're not seeing. The engineering mindset is looking for details that have a logical progression that's not in the standard model of Darwinian evolution. So what we need to do is look at this in more detail. I will not be going from a basis of creationism. I will be going from a basis of intelligent design. That is a logic that you look at the actual details. You look at the science and you build a study, a model together. And we're going to see how that fits as we go on to this area. The Salem hypothesis cuts both ways. Both they do not understand what we're saying, but if you look carefully, we do not understand some of the details of biology. We have to go as deep as we can to understand what's going on. These biologists don't understand why some engineers did not accept this narrative, this story about evolution that's been refined and polished over the past 150 years. Why not? What is there on the other side? What is the engineering perspective? What can't they see from the engineering side because they haven't studied the engineering, they haven't maybe looked at probability and statistics. So there's things that can be done there. Is there anything missing from the evolutionary narrative? What does it bring in? What are the other disciplines that it supplements? There's mathematics, statistics, and we're going to be using some simple ones. There's geophysics, which are the resources with which it works. And then you may make another step to run a simulation of a quantitative model to see how well evolution might work. Let's go back to this point that the evolution is don't follow the statistics. You can't know it all. There's too much data out there. And I think many people don't have a good understanding of how probability grows. For example, we all know that when you flip a coin, you have a 50-50 chance. Well, what if you wanted to calculate the chance of getting 10 heads? Isn't that 10 times harder? So wouldn't that be, instead of 50%, only a tenth of that, 5%? In reality, it's much less. It's about 1 chance in 1,000. If you wanted to get 20 heads, it's about 1 in 1,000,000. It grows exponentially. And that's, if you don't do the math, you don't understand this. It's not intuitive. When I look at evolutionary theory, I think it's kind of stuck where it was 150 or 100 years ago. It's a story. In a computer analogy, it's like a flow chart. You lay out what's going to happen and you show the flow going through there. But anyone who's done any programming knows there's a big difference between a flow chart and actually writing code that works. That is much more difficult. And I think that's what we have to look at here. You have to dig deep enough to understand it. Now you're probably wondering, why is there a picture of a house off to the right there? Isn't it a beautiful designed house? You can get the picture off the internet. Well, if you've bought a house, you know there's a lot of different criteria you might use to buy that house. Typically in real estate, there's a slogan: the 3 most important things are location, location, location. But a lot of people look at curb appeal. You drive up and you say, isn't that a beautiful house? I think that's kind of the evolutionary theory approach. You're looking at this beautiful story that's been developed over 150 years. When an engineer looks at a house, their interests may be different. They may say, well, wait a minute, how good is the water system? Is the electrical up to code? Let me check the basement for insects. So as a spoiler alert, I'm going to warn you that I think we're going to find some termites in the bottom of this house. The outline of our talk really covers 4 points. We need to start with, what really is evolution anyhow? Let's go through a description of it, and then we're going to look at intelligent design, irreducible complexity, which are about 20-year-old concepts. And then we're going to do actual computation, and then try and understand what this really means. So we'll start with evolution itself. Evolution has a number of different pieces. The first one we might put in, one definition, is change over time, where you have random variations, response to environmental changes. So examples there of what happens, well, we're not exact duplicates of our parents. You know, there's genetic shift. We're a little different. And of course, if you go back more generations or look at your children, there's more changes there yet. Now, that can be acted on by the environment. That's where survival of the fittest comes in. So think of an eagle. If new generations of eagle have better eyesights, they're going to be better forager. Eagles with the best eyesights will be preferred. And of course, we use that when we breed cattle, dairy cattle, for better milk production. Milk production's increased probably a factor of 2 or 3 per cow over the last 100 years. Same thing with crops. Corn production's much better. We're breeding for better survival or better features. However, the negative side of survival of the fittest is when things aren't the best. Evolution seems to have a viewpoint that things get better and better and better, but if you think of invasive species, that's the negative side of survival of the fittest. When I look at my front lawn or my garden, every year I'm fighting against things that are growing there that I don't want. That's how survival of the fittest works. I don't think anyone's ever said, you know, daffodils and tulips are crowding out the dandelions in my dandelion and thistle garden. The next step of this is many generations going on. And this is kind of like when you draw plots of a graph. you kind of extrapolate into the future or extrapolate in the back. And that's kind of what evolution does. Through many, many generations, it says you can go from simple to complex. As an engineer, I say it's much more difficult than that. You know, we can't even predict weather tomorrow or a few days from now. In Colorado right now, it's snowing. Some days ago, they weren't expecting that. So things change, and we cannot go very far out understanding what's going to happen. The progression is difficult. There's another step, and that's development of life from non-life. And even evolutionists really can handle that, because that's not really evolution. That's kind of the start. And once you have something that evolves. For our definition, we're going to take the third one, which is an undirected advancement from simple to complex. And here's a schematic picture of what we often see in those drawings. But you know, some of the supporters of evolution have a sense of humor. And so when you look at it, sometimes they realize it doesn't always go onward and better. That somehow there's degrading and regression back into the past. Let's move on to intelligent design and irreducible complexity. What are they? Well, this kind of comes out of the complexity of life itself. How do things grow? How do they start? Well, the cell itself is not a simple bag of protoplasm that, as they thought back at the days of Darwin. In other words, you can't make a jellyfish by taking a balloon and filling it with jello. There's a lot more inside there that allows it to move, to grow, to reproduce. And as time have gone on, our technology of researching with better microscopes and better techniques, X-ray diffraction, we now see what's inside the cells. There are complex machines that perform functions. In turn, those cells are made, and machines are made of proteins. The proteins in turn are made of amino acids. So there's life parts that are built upward into the large things. It's almost like a Lego approach, where when we build things, we saw things smaller, we whittle things down, we put machine shop making smaller, but life goes from the bottom up. So the 100 years of instrumentation has really allowed us to understand much more about life. Another point, inside those cells, there's instructions, the molecular machines that allow them to replicate and build more. How did those instructions come into place? Those are some of the challenges that are asked, and that's where intelligent design comes in. The world appears designed. If you were walking and found a phone lying on the ground, you would not think, oh, how did the silicon fuse in a lightning strike, and where did this come together? You'd think about who dropped it. Where did that phone come from? If you want to look at a religious point of view, here's a quote from the Bible that shows that. I think many people following intelligent design have a religious component to it, but it's not required. Intelligent design comes from the data itself. It comes from the science, but it is fully compatible with beliefs in a designer out there. In other words, they would, if you have a religious point of view, you know who the designer is. Otherwise, you're looking at design and trying to understand it. Even Richard Dawkins sees intelligent design. One of his quotes is, "Biology is the study of complicated things that have the appearance of being designed with a purpose." However, he says there is no purpose there. It's just an appearance. Whenever I see that quote, I immediately think of the movie of Wizard of Oz. And I think of the scene when Dorothy is in front of the wizard, and you hear the wizard say, "Pay no attention to the man behind the curtain." And I think that's exactly what Richard Dawkins is trying to do. He's trying to distract us from understanding, is there a purpose and what is it? Some examples of intelligent design are the climate on Earth. It's not too hot, it's not too cold, we have the right amount of water, we have oxygen, but not too much. Some of these things are put together in a movie called Privileged Planet, which really talks about how unusual life on Earth is. You know, science fiction shows all kinds of cultures and forms of life on other planets, but in reality, we haven't found anything like that. It seems like the Earth is very unique. So there is a concept of uniqueness for what we have. Irreducible complexity is something that was coined by Michael Behe, who's a biochemist, and he wrote a book, Darwin's Black Box. He uses a mousetrap analogy, which I use here because it is very compelling to me. You need all these different components. You need the block, you need the spring, you need the hammer, you need the bar to set the thing. If you had any of these missing, it's not going to work. That means you cannot incrementally design a mousetrap. If you just had the hammer, it's not going to function. The idea of evolution slowly improving on something doesn't work. This is a hurdle where you have to have all these pieces to function. Therefore, how can this happen through evolutionary framework? And then let's put it together with intelligent design. This has a very clear structure to it. If you put all these pieces in a box and shake them up, after an hour, you don't really expect to see a mousetrap. You expect to see pieces. If you shake it for a year, 100 years, multiple pieces of it, you're still not going to get a mousetrap. It just doesn't work. You really need all the parts at one time, and you need to have them organized. How does this apply to the biology and evolution? One example, the one we're going to talk about, is vision cannot occur gradually in small steps. You either have the vision or you don't have the vision. Now, if you look at a textbook or a resource on this, here's some from Encyclopedia Britannica. They've lined up about a bunch of different steps. On the far left, you've got an eye, a human eye on the far left. Then you have eye complexity and molex, where they've lined up different degrees of complexity, starting from very simple pigment spot to a pinhole lens in a nautilus, a snail. Finally, the octopus is the most complete. And they're saying, well, that's how it could have occurred. Well, let's just start with the far left one, looking right at that pigment spot there, and try and understand how a pigment spot could form from nothing. We're going to look at a single cell creature, Chlamydomonas. It's a simple alga, which is the singular of algae, and it's found in pond water. At the left is a micrograph at maybe 1,000, 2,000 times what they look like. They're little balls with a tail on them, and they move around inside the water. At the right, you see a schematic of what's inside there. It's got these tails. Here they're labeled at cilium, but probably flagellum would be a better term or more commonly used term. Inside, I want to point out two features. There's a large green area, which is a chloroplast. That's where it takes sunlight and actually makes its own energy. And the other thing I want to point out is a small area right on the side here, which is its eyespot. Eyespot allows it to see where you have light. So if you think of this as kind of a part plant, part animal, it's living in pond, and let's assume a leaf blows in and that leaf shades it from the sunlight. Well, it can't form energy. It can't perform photosynthesis unless it's in sunlight. So it uses its flagella to move until it's back in the sunlight. So it has ability to adjust and move around in that environment to get the area where it has what it needs to survive. And of course, there's a lot of other complexity. How does it make decisions? What are the other things in here that allow it to reproduce? And we're just going to skip over that. It is really like a factory, a city, because it has all these components. Let's move on to developing that capability. How would you go from a blind Chlamydomonas to one that has an eye spot, a functional eye spot? Well, of course, the evolutionary approach shows that there's a lot of steps in there. And let's look at some of these. Well, if we're going to make the proteins to work in there. There are about 20 different types of proteins that are separated into different groups that are polar, non-polar, just broad categories. But maybe a very primitive eyespot wouldn't need all of those. Maybe we could just take one from each of those different kinds. And so we could choose the key 4 spots, 4 proteins, 4 amino acids when we construct this protein, a very primitive eyespot capability. Those who have studied the Chlamydomonas structure have proteins that are 300 to 700 amino acids long. But once again, let's assume the most primitive eyespot has very short proteins, very simple chains, easier to make. And we're going to say, let's assume they're the simplest one there, and they're about 100 amino acids long. So we've tried to help the evolutionary approach move along by simplifying the number of amino acids we need to choose from, simplifying the length of the protein chain. And now let's look at how many proteins are there. Studies on the eyespot indicate there's over 200 proteins there. But one thing about evolutionary thought is you can often reuse and recycle proteins. We're going to assume that most of those proteins are already there. 192 of them are present already, and we only need 10 more proteins. So we have to put these together. Now we're going to come up with some machine inside a cell. There are ribosomes that manufacture them, but we're going to assume that we have a machine that can put those together in all the different combinations. We've got 4 proteins. We have 4 different amino acids we're going to put together in chains to make the proteins. So our first one, we choose 1 amino acids, and then among those 4, we're going to choose our second amino acid to build a chain, and we're going to keep building one after another. So it's 4 times 4 times 4 different choices. 4 times 4 is 16, times 4 is 64, times 4 is 128. It keeps growing rapidly. And we use that symbol there of 4 to the 100 power to indicate that we're multiplying 4 times itself 100 times, because that's how many different combinations there are in the first protein. But we need 10 of these proteins. That's 4 to the 100 to the 10th power, which is 4 to the 1000 power. Now that's kind of a strange concept because we don't think in multiplying by 4, we think in multiplying in base 10, but mathematically you convert that, and that comes to 10 to the 602 power. That is 10 with 602 zeros behind it. So if you're thinking about random progression here, that is a huge number of coin flips in order to chance to get this right. How big is that? It's beyond astronomical because they estimate there's only 10 to the 80 particles in the universe. So if you think of every particle being a different kind of protein, it's something that's beyond our ability to really handle that. How can we come up with that? Now, the evolutionary approach is, well, yeah, it's difficult, but it just happened. It came about. Now, those of you who don't like math, I've included a cartoon here. Mr. Osborne, may I be excused, my brain is full. We're multiplying 10 times itself 602 times. And how do we understand number? How can we accept that? Just hang with me here, and I'm going to give you some analogies of how big that number is and how to understand it, because this really is the big hurdle of evolution. How do we overcome that number? Well, I'm going to give you a chessboard and rice analogy. Perhaps you've heard it. It shows the power of multiplication. The idea is a debt was paid by putting rice on a chessboard. 1 grain on the first square, doubling it on each following square. Well, you probably know what that would look like, and we'll see how that grows. Here's a schematic of that chessboard. You see the rice grains on that first row. The farthest right would have been a pile of 128, but there'd be a pile that would be so high you can't see the individual grains anymore. So we just write the number. On the second number, a second row, we start having to put commas in because we're getting so many numbers that the numbers are getting smaller to fit in there. By the time we get down to the fourth row, we have to abbreviate thousands and then millions. So the K represents thousands and the M is mega for millions. And as we go down row after row after row, they get bigger. I have not put numbers or symbols on the bottom three rows because these numbers are so big, they're kind of it's understandable. I mean, how do we recognize those abbreviations for such large numbers? Those are numbers like the national debt in dollars. It's a number that's hard to comprehend, or maybe you can think of it as the number of Starbucks in Denver. How many are there? It's hard to imagine how many there are. To help understand these numbers, I made a table showing what's on those different rows and adding them up. On the first row, there is 256 minus 1, so 255 grains. That's a few grams and would fit in a tablespoon. If we put the second row together, now we're in 64,000 grains of rice. It's about 4 pounds, and that would fill in a gallon container. Now, if we look up to the third row, now we're in millions of grains of rice. It's about a half ton. That's equivalent to a pickup truck load. Fourth row takes us into billions with over 100 tons of rice, and that would fill up 2 freight cars. Fifth row goes to 1 trillion grains of rice. Now we have to put that on a barge. Sixth row, we're in hundreds of trillions, 8 million tons of rice. It fills up 20 ocean freighters. Seventh row keeps going up. Now we up 2 billion tons of rice, which is about the world yearly production. Finish up with the eighth row here. Numbers that are kind of meaningless. I'll just talk about volume. It covers the county we live in, 150 foot deep. It's an amazing number. How can we understand it? In fact, that is probably equal to all the rice that's ever been raised in the history of the world, because years ago it wasn't cultivated to the level it is now. So that is just one example of it. Let's go back to the eyespot. The ways to make those 10 proteins is much, much, much larger than the rice on a chessboard. The 2 to the 64 is roughly equal 10 to the 19th. So doesn't that sound like it's virtually impossible to form by chance? How could evolution explain this? Oh, wait, we have the whole age of the Earth for this to happen. And instead of a simple one organism, we might have a whole pond full of them. So let's see how that speeds things up and how well it works. Now let's calculate how this works. You probably remember there's an equation how we can put this together, but evolution is going to need some help here. So instead of just a pond, let's assume all the oceans, all those molecules are available to make the proteins. And we're going to assume that 10% of the water is amino acids. Now we're going to put those amino acids together at the fastest rate that cells can form proteins, which is about 1,000 times a second. And we'll keep moving on there. How long will it take to go through all the combinations to make the 10 right proteins? This is where a simple equation of rate and speed comes in. Oh, and we're going to ignore the problem of getting all these proteins into the right place in that single organism, because if we're all in oceans, maybe one protein's in the North Atlantic, one's in the South Atlantic, one's in the Indian Ocean, so on. Well, let's do our calculation and see how long it takes. If you take the number of permutations, which is that 10 to the 602, and divide it by the rate, which is how many of these there are working at whatever rate, that'll come up with the answer in time. Let's do that calculation. So it's 10 to the 600 combinations. And there we have our 10 to the 46 amino acids in all the oceans, reacting 10 to the 3 or 1,000 times a second. There's about 10 to the 7 seconds in a year. And the answer comes out to 10 to the 540 years to make this work. Therefore, an age of the Earth of even 4.5 billion years is woefully inadequate to development of even the few proteins we need for this eyespot to function. Also, the information stored in the machinery is like the information encoded in computer programs. How did this appear? How do we get the information in there to make this work? Well, what's left, the best remaining mechanism is intelligent design with a handprint of a designer. And you need to look for that designer. If it's indicating there's one out there, how would you find that designer? What does this really mean? Well, the likelihood of forming these proteins by chance is similar to winning the lottery 60 times in a row. Now, I think all of you would say, well, you know, someone's going to win a lottery. Why can't it be me? And that works. I mean, the first time if you win the lottery, you're going to get your article in local paper, you're going to be interviewed, you're going to get 100 letters from financial advisors who would like you to help you to handle that money. And also, you'll be getting a belated birthday card from your cousin Louie, who you haven't seen in years, saying, hey, how you doing? What happens when you win this lottery the second time? Now, the lottery is going to do an audit because they know it's virtually impossible for you to win a lottery a second time. You're going to get an article in the New York Times, and you're going to get 1,000 letters from financial advisors. By the time you win the lottery the third time, now you're going to make the front page of the National Enquirer. Babies are going to be named after you. People are going to be asking for advice on numbers. By the fourth time, you're going to start getting death threats from people who are saying, wait, it's my turn to win. You can't just keep winning. You're going to be prohibited by law. They're going to pass an amendment to the Constitution that says you can't play anymore. By the fifth time you win the lottery, no one's going to buy tickets anymore. The lottery is going to go bankrupt. It's going to be impossible, but yet there's another lottery. Well, when you win the lottery again, pretty soon the Earth tilts off its axis. It just can't keep going like this. So winning the lottery that many times is impossible, which takes us there to undirected development of complexity, even for an eyespot, is not science. It's faith, and it requires more faith in Christianity. And since we're talking about eyespot, can't we just call it blind faith that the evolutionists have? And yet, in spite of this, they claim there's no credible arguments against evolution. I would submit this is a credible argument, and I have not had anyone comment with a solution to this or an alternate explanation. Let's look more at what this means. Some people say the Earth is 6,000 to 10,000 years old. Some people say it's 4.5 billion years old. This is how many years it would take for development of this eyespot, functional eyespot, or a random chance. Look at that comparison. People who argue over the age of the Earth between those first two spots are missing the point. They're missing the complexity of life and evolution viewpoint. At this point, I want to just acknowledge some help. My wife is a biostatistician, and she's reviewed this to make sure I wasn't making any strange comments and getting off track. So that has helped me a lot to give me confidence that this is right. I did get images and various other things from Wikipedia and eLife. And I also want to point out Michael Behe. I used his analogy of the mice trap, which brings me to some references. My wife and I did write a short summary of this several years ago for a magazine called Salvo. So if you wanted to see what we've said here in a shorter, maybe 3-4 page version, you'd be able to look that up. The second bullet there is a true scientific write-up of this, which goes into a lot more detail of the statistics, background information, etc. And it's a journal, Bioinnovation, that you can find online. It's free access. The third one is I give you a little more data on Darwin's black box. I think he has some other analogies of the complexity in there of blood clotting. How could a flagellum form? So he really makes the situation even more complex for that. And let me go back to Privileged Planet again, because I think that goes into the cosmology complexity of intelligent design. And I am working on a talk on that will be given sometime in the future. And then finally, I just point out the store at the Discovery Institute website, which has more resources and books on intelligent design. And that finishes my talk, and now I'll pass it over to David, who can give me some of the questions that have been listed. David?
Okay, one of the questions was you only calculated the number of possible combinations, but the question is how many combinations might be helpful to the organism in some way?
Well, that's a very good question. And I think a number of the other combinations would be useful. I mean, the Chlamydomonas has many, many of those proteins inside there. But these are the ones that have a function. These are the ones that are in an active eyespot. I think you probably need to choose them, but let's just say 1 billion combinations of these are interchangeable. That means you would divide 10 to the 540 years by 1 billion, and you still come up with an amazing number.
Yeah, you might want to refer to some of Doug Axe's work if you're familiar with that. Might also be a good answer for that question.
That's true. And if you go to the Discovery Institute website, you can find Doug Axe's book, which is Undeniable. And he goes through calculations of protein folding in there, how complex it is, because these, I didn't talk about the fact of the different types of proteins, but they do have the different types of proteins have to be attracted to one another with hydrogen bonding to fold properly. So again, we only chose 4 amino acids out of the 20 that are in there. And we only chose 100 long proteins, where in an actual eyespot they're 300, 500, much, and of course those numbers then get much, much larger again. So I think we've been conservative. I don't think we're really swaying it to the benefit. I think we've tried to go to the evolution side and give them more time and more liquid and more amino acids, and it still comes up with an amazing number.
All right, if other folks have questions, if you want to type them in the chat, or if you just want to raise your hand and ask the question, you can do that as well. I'll be looking for that.
Meanwhile, I'll mention that I believe this has been recorded, and we will be finding a link to that and making it available to those who want to maybe pick up the first part if they missed it, or if you want to share it with someone else. Also, we do plan to have more of these talks in the future, and you will be getting emails about more talks as time goes on.
We had the question that, it was a really good question, how do evolutionists address this high probability? How do they address the whole probabilistic resources problem? What's their response to it?
I haven't seen a good answer. I have not seen any evolutionists come up with a different model of that. It tends to be more descriptive model. It's what some people would call a hand-waving argument, say, well, there's lots of things happen and there's certain environment where it would work better, but I really haven't seen something that's quantitative, that's more of the design approach. So I guess if there's anyone who knows someone who has an evolution background, let's propose a counter model. I'd like to see it.
Yeah, I will have to say that most of the responses that I have seen have been either just dismissal, just outright dismissal, but the ones who have tried to take it on have basically just asserted that selection works even better than we thought it did, with no evidence for that, right? So that's it. We have another question here. What about parts of life that seem not designed, like the recurrent laryngeal nerve that in all mammals that pass down the neck near the heart and back up the neck, even in a giraffe? Which may be all, they said that may be all off topic, but it's a good question.
Okay, maybe to build on that a little further, let me take you down to that second bullet, because that really addresses that actual question. If you look there, we raise it and we challenge them, saying there is a need for a quantitative model. Can you counter what we see here? If it really does happen that way, put something together. So I would say that second bullet is kind of a challenge to the evolutionists to come up with something that would work better. And while there might be a thought about the next question here, let me just point out in 2 weeks, we will have another talk. Paul Ernst will be giving his talk on this same series. Unfortunately, I don't have it on my screen because I am using this screen to give you these resources, so I can't quick switch to my other screen and bring up the announcement, but you will be getting another email. It'll be Monday in 2 weeks at 7 o'clock Mountain Time.
Okay, we have one other question, a really interesting question on this line here. It says, hi, I'm a molecular biologist. I'm working on characterizing proteins involved in metastasis. Some of the work that I did, was based on in silico work that utilized advanced algorithms to determine protein structure and folding. I'm not a coder or a statistician, but wouldn't the developers of these programs have to understand the probability problems? If clarification is needed, please let me know. And Tom, you can correct me on this if I'm wrong, but I think you're pointing out the search space, the size of the search space that Tom has been talking about is, and Doug Axe talks about, is so enormous because there's nobody in the evolutionary model, there's nobody doing that programming. Of course, you can winnow down the number of possible choices in search spaces if you have an intelligent person writing the algorithm. But if there's nobody writing the algorithm, if it's just reliant on natural selection working on, again, a random mutation, the search space is vast. So yeah, they start off with a very large search space, but write the algorithms specifically to limit it to things that will essentially work. Tom, is that?
Yes. Well, let me back up to one thing, because I was also advertising a talk in 2 weeks by Paul Ernst, and I do have the title now. It's Bad Design is Still Design. So it's really a case about optimization and how do you get something to work. Okay, let me go back to the question you're asking here. You really do need to consider protein folding, which is not in this model, and that does need to be covered in there. But these things are very complex to model. I would guess one reason that it's not covered by researchers in this area is it's very hard to put this together and come up with something that matches reality. There's so many variables, it's difficult. Also, I think if people have tried to do this, they're going to come up with answers they don't like, which means they're not going to want to publish it. And if they came up with something that others didn't like, no one would publish it, even if they wanted to publish it. I think part of the problem is they're really hitting a dead end. They're kind of going inside a horseshoe canyon, and there may not be any way further forward on this. So yes, those who are looking at this should consider the various permutations and combinations as much as they can. And so I encourage people to go forward, making something a little more complex than what we have here, as complex as they can, that still allows us to come together and come up with some data. But it's going to come up with, again, incredible numbers. I cannot imagine coming up with something that says this works, that evolution works.
Okay. We don't have any more questions here coming in at the moment.
Okay, well, people have other things to do, so I would just say thank you for joining us tonight, and we'll see you in 2 weeks for the next talk on design, sponsored by SCN Colorado. Thanks again.
Thank you, Tom.
Thank you, Tom. It was terrific.
Glad you enjoyed it. And again, my screen's kind of busy here with the various things going on, but I do see the recording button is on now that I've minimized sharing screen. So that should be available, and we'll send out the link to this and you'll have a chance to look at it again. Thanks for attending.
Thanks so much.
Tom, I have a question. This is
Oh,
Donald.
yes, go ahead.
I'm not a statistician at all, but I was just wondering, I've heard pushback on just the use of stats anyway, is that you have to know all the parameters about every aspect of whatever you're trying to derive your statistics. Have you heard that pushback? You know, I don't know what they would apply it to, maybe the inside of a cell or whatever, you know, if you're trying to create a cell or just a protein or whatever, you have to know everything about it to really get accurate stats.
Well, we have not had any pushback at all. I think part of it is this is a very simple model. There might be maybe more comments if we developed it with more structure, but the more we develop it, the harder it is to explain. But I also think it's going to show that evolution is even less likely. So I don't know that much needs to be done until we get a counter argument from someone supporting evolution.
Okay. Okay, I can just see the counter being not even dealing with evolution, just saying attacking the statistical probability of things.
I would agree. I think you ignore this because you don't have an answer. You know, sometimes old jokes used to be, if you don't like the message, kill the messenger or ignore the messenger. I don't think there is a good response to this. We've tried to simplify this as much as we could in putting it in it, because see, we've taken the better part of an hour to cover it here. If we try to expand it more, it's going to be more difficult to explain, and it's going to be even less probable when you put in those other constraints on it. So I think this is as far as we need to go right now.
I see. All right. Thank you very much. I enjoyed it.
Our pleasure to share this. And as I said, there'll be more coming in the future. So we hope to have a series of these by the Colorado chapter.
All right, everybody, that's a wrap. We'll see y'all later. Have a good evening.
Thanks, David, for your help in putting this up for us. Thanks. We'll see you in 2 weeks.